Dear students, in today's lecture, let us know about status of mulberry diseases and their classification. Mulberry morus species, the sole food plant of Silkworm, Bombyx mori, is cultivated in both tropical and temperate countries under wide range of ecological conditions. During its growth, like any other crop plants, various types of pathogens such as fungi, bacteria, viruses, mycoplasma and nematodes affect mulberry. In fact, diseases are regarded as one of the major constraints for successful mulberry cultivation and production of quality leaves. The diseases, in addition to the reduction in leaf yield, also affect the quality of the leaf. Biochemical studies have revealed that the diseased leaves are poor in nutritive values and moisture content. Feeding such leaves not only affects the growth and development of silkworms, but also the cocoon yield and silk quality. The intensity of diseases varies with season, variety and cultivation practices. Also, being a perennial crop, the pathogens seem to readily perpetuate in mulberry gardens throughout the year and quickly spread to cover extensive areas. However, it is primarily the fungi that cause greater damage to plant health and crop productivity in mulberry. The fungal diseases are more predominant and cause 10 to 15% loss in leaf yield, besides deteriorating the leaf quality. Symptoms and signs in diagnosis of mulberry diseases. Disease is a series of harmful physiological processes in which the plants become abnormal by the infection of causal agents or environmental factors, which leads to development of symptoms. The diagnosis is one of the most important and useful criteria to recognize the identity of a disease that helps to save the plant from disease by adopting suitable control measures. The role of symptoms and signs in diagnosis of any disease are different. The disease is first noticed by the presence of symptoms. The symptoms indicate the form of occurrence of diseases in the form of spot or wilting or blight, etc., whereas signs can point directly to the causal agents that can suggest whether a disease is infectious or non-infectious in origin. During diagnosis of a disease, examination of underground parts of the host plant should not be neglected, because many symptoms that express above the ground may also be due to a problem with the roots or base of the stem. The microbes which have the capacity to cause disease in the host plants are known as causal agents or pathogens, while an organism that harbors or supports the activities of a pathogen is known as a host. When more than one causal agents cause disease, it is called disease complex. The external and internal alterations visible on the plants due to disease are called symptoms. The presence of pathogen spots like vegetative, reproductive structures, mycelium and spores on a host plants is termed as sign. When diseased plants have the signs of a pathogen along with symptoms in infected portions, it is known as syndrome. The study of symptoms of disease and signs of pathogens for the diagnosis of plant diseases is called symptomatology. Status of mulberry diseases. Foliar diseases. Mulberry is mainly affected by 5 foliar diseases. They are leaf spot, leaf rust, powdery mildew, leaf blight and bacterial blight. Leaf spot disease. It is one of the major diseases of mulberry caused by a fungal pathogen Cercospora moricola. This disease is common in rainy and post-rainy seasons, July to December, in South India. The disease starts progressing 35 to 40 days after pruning or leaf harvesting and becomes severe on the 70th day after pruning, when leaves are harvested in bulk. The symptoms of this disease are irregular brownish spots surrounded with chlorotic rings. And as the disease advances, the spots enlarge and coalesce and the dead tissue fall off, resulting in short hole development. This disease causes 10 to 12% leaf yield loss, besides reduction in nutritive value of leaf, especially chlorophyll, leaf moisture, protein and sugar contents. Management. Cultural control. Wider spacing of 90 by 90 centimeter or paired row planting systems 90 plus 150 into 60 centimeter should be followed. Chemical control. Bavestin or carbendazim 50% WP solution at the rate of 2% concentration is recommended, which reduces up to 70% of the disease. The safe period to use the leaf for feeding of silkworms is 2 to 3 days. Powdery mildew. This disease is one of the major foliar diseases caused by Phylloxera corylea, which is also known as Phylloxera moricola. It is more prevalent in tropics and temperate regions during post-rainy and winter seasons and starts progressing 40th days after pruning. The symptoms of this disease is white powdery patches on the lower surface of the leaf, and as the disease advances, the entire leaf turns to brownish black and fall off prematurely. The loss due to mildew is around 5 to 10% annually, besides causing depletion in nutritive value. Management. Cultural control. Wider spacing or paired row planting system should be followed. Chemical control. Spray 0.2% Bavestin or 0.2% keratin on the lower surface of the leaves. Leaf rust. There are two types of rust diseases which are common in mulberry. They are black rust and red rust. Black rust is caused by Ceratolium fizzi and also known as Peridiaspora mori. The disease is prevalent during winter and rainy seasons. The symptoms of black rust disease are circular pinhead-sized blackish brown spots. It is an obligate parasite and starts progressing 45 to 50 days after pruning. The mature leaves are more prone to the disease, and 10 to 15% crop loss is estimated annually. Red rust is caused by Ascidium mori and commonly observed in Himachal Pradesh, Uttar Pradesh, West Bengal, northeastern region, and also to some extent in Karnataka. This disease causes up to 15% leaf yield loss. Red rust affects young buds, leaves, petioles and shoots. The symptoms are young buds become swollen with upward curling and with protruded golden yellow spots. The fungus spreads through vascular bundles. Management. Cultural control. Avoid delay in harvesting of leaves. Wider spacing or paired row planting system should be followed. Chemical control. Fungicide 0.2% Kavach, that is chlorothalonil 75% WP, is recommended for black rust and 0.2% Bavestin dusting for red rust. Fungal leaf blight. This disease is caused by various species of fungi, Alternaria alternata, Fusarium pallidorosium. It reduces up to 10% leaf yield loss with depletion in nutritive values. The symptoms of the disease are blackening of leaves from the tip or edges of lamina. As the disease advances, the entire leaf surface is affected and fall off prematurely. It is more prevalent in tropical regions. Management. Cultural control. Avoid delay in harvesting of leaves. Wider spacing or paired row planting system should be followed. Chemical control. 0.2% Dithanium 45 has been found effective against leaf blight. Bacterial leaf blight. It is caused by Pseudomonas syringae. This disease is more common in rainy and winter seasons, and it causes about 8 to 12% leaf yield loss. The symptoms of this disease are numerous water-soaked spots which deteriorates nutritive value. Management. Cultural control. Avoid delay in harvesting of leaves. Wider spacing of 90 by 90 cm or paired row planting system of 90 plus 150 into 60 cm should be followed. Chemical control. Agricultural antibiotics such as 0.2% streptomycin or 0.2% tetracycline are effective. Root diseases. Number 1. Root rot. Due to its epidemic nature and potential to kill the plants, root rot is regarded as a dangerous disease of mulberry. It is widely distributed in almost all the sericultural areas, occurring in all types of soil throughout the year, resulting in 30% mortality of plants with 14% reduction in leaf. It is caused mainly by Rhizoctonia botasticola. This disease spreads at a faster rate in soils with 30 to 35 degrees centigrade temperature range and moisture level of less than 40%. The disease is characterized by sudden wilting of leaves followed by withering, decaying of roots and death of the affected plants. The disease initially appears in few plants as isolated patches, which acts as infective centers, leading to mortality of well-established plants within a short span. As the disease progresses, secondary fungi such as Fusarium species, Botrydiplodia theobrome, etc. colonize and decay the roots. Management. The earlier methods of using fungicides such as Indophilium 45, carbamates, fumigants, organofungicides as well as Raksha, that is Trichoderma harzianum, did not provide desirable control. Hence, an eco-friendly technology has been evolved at CSRTM Mysore with the invention of Navinia, which contains 80% plant products and 20% chemicals. Method of application of Navinia against root rot. Prepare Navinia solution by adding 10 grams Navinia in 1 liter water and stir well. 1 kg Navinia per 100 liter is sufficient for 100 plants. Prune the dried or wilted plants 15 to 30 centimeters above ground level. Make a shallow ring basin around the plant stem to avoid runoff. Pour 1 liter solution over the pruned stump to drench completely up to the ground level. Treat the surrounding plants also to prevent spread of the disease. Precautions. Do not irrigate the treated mulberry plants for 4 to 5 days. Remove dead mulberry plants and destroy by burning, and also expose the soil to sunlight. Replant with new saplings after dipping their roots in 0.4% Navinia solution for 15 minutes. Maintain optimum organic carbon content of more than 0.5% and soil moisture above 40% to prevent this disease. Number 2. Root knot. Another major root disease is root knot caused by a nematode Melodagine incognita. This disease is characterized by the galls or knots on the root system, which is characteristic symptom. It results in stunted growth and reduced yield, and many times it makes a way for the entry of root rot causing pathogens, resulting in death of plants. Leaf yield loss due to root knot has been reported to be 10 to 12%. Management. To control root knot disease, Nemahari, an eco-friendly product, is developed by CSRTI Mysore. Method of application. 2 applications per year if less than 50 galls per plant are found. 3 applications per year if more than 50 galls are found per plant. Dosage of application. 40 kg per hectare, that is, mix 40 kg Nemahari with 400 kg FYM or compost. First application, Make trenches of 15 cm deep near the root zone. Apply Nemahari mixture and cover with soil followed by irrigation. Second application 70 to 80 days after first application. Third application 40 to 150 days after second application. Root disease complex. Besides root knot and root rot diseases, root disease complex has become a major production constraint in mulberry in recent times. It is caused by the association of root knot nematode Melanogain incognita and root rot fungal pathogens, that is Fusarium solani, Fusarium oxysporum, Botrytis theobrome, etc. When the root system is severely infected with nematode, it makes good site for easy entry of root rot pathogens. Severity of disease results in the death of mulberry plants. This disease is characterized by wilting or burrowing and defoliation of leaves from lower portion of the branches progressing upwards, and wilting occurs in whole plant. Entire root system decays and lose grip in the soil. Roots show more than 100 galls with decayed or rotten roots. The conducting tissues become black or brown due to rotting of the tissues as the disease progresses. Crop loss is estimated to be about 15% due to this disease. Nursery diseases. Stem canker. This disease is caused by a fungus, Botrytis theobrome, which is characterized by greenish black eruptions on cuttings. This is a major problem in nurseries which causes rotting of the whole cutting. This disease causes around 40 to 45% loss. Cutting rot. This is caused by Fusarium solani and appears as rotting of the whole cuttings and decaying of bark, resulting in death of the sprouted cuttings. Collar rot. This is caused by Phoma sorghina or Phoma mororum and appears as brown to black discoloration of bark and rotting of cuttings near the soil surface line, collar region of the cuttings. Dieback. This is caused by Botrytis theobrome and characterized by the wilting of saplings at the apex and progressing downwards, resulting in death of saplings. Crop loss due to nursery diseases. The nursery diseases affect the sprouting of cuttings and survival of the saplings. The mortality is up to 35%. The mortality may be still high, that is 50%, in the case of high yielding poor rooting varieties. Management. Cultural control. Plowing the land deeply and exposing the soil to sunlight for about a month, leveling to avoid waterlogging and keeping land free from weeds during establishment of saplings or plants. Chemical control. Treating the cuttings or dressing the stem cuttings with 0.1% dithionium 45 is recommended. Integrated management. This method involves dipping of cuttings in 0.1% dithionium 45 solution and application of bioformulation called Nursery Guard, a product of CSRTA Mysore, prepared by using Trichoderma pseudoconingi. Classification of mulberry diseases. Mulberry diseases can be classified on the basis of mode of spread, prevalence and periodicity, plant parts affected and parasitic nature, etc. Classification based on mode of spread. According to mode of spread, mulberry diseases can be categorized into three groups such as seed-borne, soil-borne and airborne diseases. Seed-borne diseases. The diseases which transmit through the seeds are called seed-borne diseases. Leaf spot of rice, ergot disease of bajra are the examples of seed-borne diseases. In mulberry, there are no seed-borne diseases. Soil-borne diseases. The diseases which spread through the soil are called soil-borne diseases. The examples of soil-borne diseases in mulberry are root knot, root rot, collar rot and cutting rot. Airborne diseases. The diseases which spread through air are called airborne diseases. Most common examples of airborne diseases in mulberry are leaf spot, powdery mildew and leaf rust. Classification based on prevalence and periodicity. According to prevalence and periodicity, mulberry diseases can be categorized into 4 groups such as epidemic, endemic, sporadic and pandemic diseases. Epidemic: the disease which occurs periodically in widespread areas, causes devastating damage to the crops and spreads very fast from one place to another, is called epidemic disease. Examples of epidemic diseases of mulberry are leaf rust and root rot. Endemic: when a disease is constantly present in a locality and occurs year after year, it is known as endemic disease. Examples of endemic diseases of mulberry are leaf spot and root knot. Sporadic: the disease which occurs at irregular intervals over a limited area is called sporadic disease. Examples of sporadic diseases of mulberry are leaf blight and black leaf spot. Pandemic: a particular disease that occurs all over the world and causes heavy damage is called pandemic disease. Examples of pandemic diseases of mulberry are root knot and root rot. Classification based on mode of spread. Based on the plant parts affected, the diseases of mulberry can be classified into 4 groups such as foliar diseases, root diseases, vascular diseases and systemic diseases. Foliar diseases: the diseases which appear on above ground parts of plants, especially foliage, are known as foliar diseases. Examples: leaf spot, leaf rust, powdery mildew. Root diseases: the diseases affecting root system are termed as root diseases. Examples of root diseases of mulberry are root knot and root rot. Vascular diseases: when the pathogens attack vascular tissues, that is xylem and phloem, and cause diseases, they are called vascular diseases. Examples of vascular diseases of mulberry are bacterial wilt and root rot. Systemic diseases: the diseases which are caused by the pathogen that spread throughout the plant system intracellularly are called systemic diseases. Examples: dwarf disease and mosaic disease of mulberry. Classification based on parasitic nature. Based on causal factors, plant diseases can be broadly classified into 2 groups, namely non-parasitic and parasitic diseases. Non-parasitic ones are also known as non-infectious or non-pathogenic or physiological diseases. Non-parasitic diseases: the diseases which occur without the involvement of microbes and are mostly caused either due to deficiency or excess of nutrients or physical and chemical injuries are known as non-parasitic diseases. Such types of diseases can be recognized only through symptoms that appear on the plants. These diseases cannot be transmitted from diseased to healthy plants and therefore are also known as non-infectious diseases. Factors responsible for occurrence of non-parasitic diseases are soil moisture imbalance. Low soil moisture causes stunted plant growth and plant bears pale green to light yellow leaves. If drought is continued, the perennial plants get defoliated and finally may wilt and die. High soil moisture results in diminished oxygen in the soil, which affects the root growth. Plants become succulent, which increases their susceptibility to certain pathogens. Then nutritional imbalance. The symptoms of nutritional imbalance mainly occur due to deficiency of macronutrients such as NPK and micronutrients such as calcium, sulfur, zinc, magnesium, etc. in the soil. The symptoms of nitrogen deficiency appear as yellowing of leaves, more particularly of leaf veins, followed by poor growth of the plants. While marginal scorching of leaves at younger stage of plant indicates the deficiency of potassium. Interveinal chlorosis followed by marginal necrosis on older leaves is the symptoms of phosphorus deficiency. With regard to micronutrient deficiency symptoms, calcium shows chlorosis followed by death of leaf tips and margins as well as rolling of leaves. The deficiency of sulfur causes yellowing of leaves with subsequent abscission at later stage. Younger leaves are more affected than the older ones. Zinc deficiency induces mottling of leaves with prominent chlorosis towards veins and whitish spots on the lamina. The iron deficit plants exhibit chlorosis of leaves with dark green veins followed by interveinal necrosis and marginal scorching. However, magnesium deficiency results in yellowing or chlorosis of leaves. The leaves may also become reddish and show necrotic spots. Then meteorological or climatic factors. Meteorological factors such as higher temperature, deficiency of light intensity, wind and storm cause various types of symptoms on host plants. Higher temperature causes sunken brown areas on plants, which leads to scorching and rolling of leaves, while light deficiency causes stunted plant growth and reduction in leaf size. The action of wind and storm results in injury and wounds in plants, which makes plants more prone to pathogen attack. Then incorrect use of pesticides and chemical fertilizers. Due to non-judicious use of pesticides and chemical fertilizers, dark blackish brown necrotic burning types of symptoms appear on different plant pots. These symptoms are exactly similar to leaf blight, but can be identified under microscope by the absence of pathogens on host plants. Then disorders caused by atmospheric impurities. Another group of disease symptoms noticed extensively in plants is caused by air pollutants, which are released by various factories or industries in the form of gases and smokes. The influence of these pollutants on plants is varied and extensive. Gaseous forms such as coal gas and smoke released from coal industry are injurious to plants, causing browning of leaf blade, premature shedding of leaves, and sometimes death of the plants. Similarly, fluorine or fluorides developed from the waste products in ceramic and fertilizer industries cause marginal necrosis to plants. Ethylene produced by burning of natural gas adversely affects the plant growth, followed by leaf dropping. Then parasitic diseases. Diseases caused by various types of pathogenic microbes such as molds, fungi, bacteria, virus, MLO, nematode, etc. are known as parasitic diseases. These diseases can be recognized by the presence of specific symptoms and signs on the host plants. Based on the infection, the symptoms can be classified into two ways, that is primary and secondary. Primary symptoms appear immediately after establishment of pathogen, whereas secondary symptoms appear in distant or uninvaded parts of the plant. We have already learnt few parasitic diseases caused by fungi and bacteria. Now let us know in brief about diseases caused by virus and MLOs in mulberry. Viral diseases. Important viral diseases of mulberry are mosaic and yellow net vein disease. Mosaic disease is caused by mosaic virus and mostly observed in temperate conditions. In India, it has been reported from Kalimpong, West Bengal. However, this disease is of less importance in sericulture industry as it does not cause any economic damage in mulberry leaf production. Yellow net vein disease is caused by a yellow net vein virus and is transmitted by the whitefly Bemisia species. In India, this disease has been reported from Kalimpong and Jammu. Control. Diseased plants should be uprooted and destroyed. Mycoplasma disease or dwarf disease. This is a very common disease in China, Japan, Korea and India. It is caused by MLO, mycoplasma-like organism. The MLO is the pellet-shaped and about 80 nanometer in diameter. The infected plants become extremely stunted. Leaves become crumpled extremely small and lose green color. The shoots become extremely slender and small with shortened internodes. This disease reduces approximately 70% of leaf yield during its epidemic condition. Control: diseased plants should be removed and destroyed. Mulberry is prone to attack by diseases such as foliar, root as well as nursery diseases, which cause considerable loss both qualitatively and quantitatively. Diseased leaves are poor in nutritive values and moisture content. Feeding such leaves not only affects the growth and development of silkworms, but also the cocoon yield and silk quality. It is primarily the fungi that cause greater damage to plant health and crop productivity in mulberry. The fungal diseases are more predominant and cause 10 to 15% loss in leaf yield, besides deteriorating the leaf quality. Integrated disease management practice involving cultural, biological and chemical measures have been suggested for the control of various diseases in mulberry.